So yeah, thanks for the invitation. So it's a pleasure to be here and be able to also to interact with those of you who actually test variants. We are only doing modeling. I love starting this talk with this good old quote from Eric Lander. He was just after the first draft of the human genome was sequenced and stated, part the book, hard to read. Many of us are trying to relate genetic genome and genetic variations to phenotypes. And that is stated like this, a great problem for, let's stay here, for supervised learning. Or can you cut this one?
No, it was necessary. Cut it at the back.
Let's cut it. All right, good. So this is a great setting, a priori for supervised learning, where we can predict phenotypes from genotypes. However, the gap is huge, and actually the starting sequence space is extremely large. And so this, in many situations, we don't have enough data to train such models in a purely supervised fashion easily. The machine learning, one of the big revolution in the recent deep learning, has been explained very well by Patrick before, and this is the idea that in many situations, just having a lot of unlabeled data, that means just having a lot of input without necessarily the measurements of what you want to predict, already allows you to actually train in a non-supervised fashion features that are then helpful for predicting what you want to predict. And this approach is called self-supervised learning, where we basically turn the problem in identifying these features by simply masking or hiding parts of the input and predicting it from the rest of the input. So this is basically learning features that capture how heights distribute. It's fitting distributions in the input data. And for us in genomic, well, we can leverage the fact that evolution is constantly mutated and then selected in particular context elements. And so we have indeed under or over representations of elements on the genomes we see, depending on where they would operate, where they will be selected. And this can be exploited. And it's a very old idea. It's the concept of comparative genomics. You compare genomes and you identify elements that are overrepresented in some sublocations. Now with the number of genomes we have, and with very scalable algorithms and techniques, we can make this in a very efficient way. So it is important when you move from coding to non-coding areas that you want to overcome limitations of alignment. And so a traditional way of in comparative genomics has been to align regions. And here I'm highlighting one particular example where we're looking at the sequence. Let me stay here. I don't know. Where we're looking at the sequence, well, this is right.
It's not you, just some people.
There's a little Lawson. All right. Can you cut one of the two, maybe? That's good? Yeah. All right. Okay. So we are looking here 3' of coding region. The region of the gene CBP3. And we've been using alignments for aligning this 200 nucleotides starting from Cerevisiae to other fungi. And what you see is that you can align the sequences as long as you stay in the genus of Saccharomyces, but you are not able to align later on. There is a motif here which is recognized by an RNA binding protein, PAF3, and that helps the degradation of those RNAs. And this is functional in Cerevisiae. It's been well studied. You would see it by alignments at similar location, but you also, if you simply scan this region with this motif, you will find it in other species where the alignment actually fails. And there's even functional evidence in CRASA that this motif indeed is still functional. So we have for those regions actually a presence of elements, maybe not at the same locations, maybe these things have been lost and reoccurred somewhere, or the actual order matters less, and so alignment is not the best way to capture those things. So we and others have been exploring the use of large language modeling in the context of DNA. A slight difference to what Patrick showed. Patrick showed the type of next token predictions where you have a piece of DNA and you predict the next token. Maybe a perhaps more general way is to do mass language modeling where you you mask at any type of positions in your input of DNA. In the beginning of this talk, I will highlight a first paper of us where we've trained a model across species that are very far from each other. And so the trick to allow the models to train from models from genomes where the code may have evolved was for us to bring another token that label from which genomes the sequence was coming. And we call this model a species LM. And otherwise, it's a classical transformer that would predict for a masked nucleotide the probability, give a probability for each of the 4 bases. We trained that across 800 fungi that are about 500 million years of evolution, spanning about 500 million years of evolution. And we devised in this first paper a strategy where we'll enrich for regions that are pretty dense in non-coding functional elements. It is pretty easy to map where coding sequences are in those genomes. And then what we decided to do is to focus on the region that is upstream of the start codon and a region that is downstream of the stop codon. We call this the upstream model, the downstream models. There were 10 billion bases. We could train model with hundreds of millions of parameters. And when we did that, what you can do is you can run the model in a particular region and then ask what is the most likely base at each position and contrast this to the random frequencies of nucleotides. Nicely, you see that there are motifs, such as here, the motif for this transcription factor that shows up, and which makes sense in the context of here, these ribosome genes that indeed is regulated by transcription factor RAP1. Our regions are blunt, it's just 1,000 nucleotides. We are not caring too much about annotations. So often we end up in a neighbor gene, and here this is the case. You end up in the 3' UTR of another gene in tandem with this gene, and now you have other motifs, which makes sense in the context of the UTRs, which are RNA binding motifs showing up. Okay, it's more than that. It's much more than discovering motifs. Actually, this model seems to model even where those motifs are functional. And to highlight this, I'll show you here data for transcription factor for which there is binding data in yeast. So this is TBF1P. And you can scan the genomes with these motifs. You will find a lot of it. Only those blue ones here are actually bound by the transcription factor. And very nicely, the models actually reconstruct with a lot of confidence those that happen to be bound, even though these models have never seen any chip data. It's just by masking and reconstructing. So this is quite encouraging, and that shows that these models not only learn words, but indeed the entire grammar. And just to, yeah, okay. So going a bit even further, you guys are doing mutagenesis scanning. We wondered whether this could be a good way also to predict what happens in situations where we're not looking at endogenous sequences, but we're looking at mutated sequences. And we revisited the data sets from Aaron Segal, who selected 200 UTRs and then performed a mutagenesis scanning with a design in which you had 10 base pair windows in which random variants were, random nucleotides were inserted. And so on this data, you can use the representation that the model does and then perform a simple linear regressions on those so-called embeddings. And we get on held out genes very good performance in terms of predictions. So that shows that these models have a good representation of endogenous sequences and are also helpful to model sequences that deviate, at least not too far, from the endogenous sequences. So for variant effect predictions. Okay, you will read in the literature since the very first DNA language model paper from 2021, many papers that do exactly this strategy. You train, and then you fine-tune on new tasks, and we have these types of pretty boring tables to show. But we wondered maybe if we could see a bit more biology that is underlying those models. And we asked this question, what is the sequence context that these DNA LMs capture? And Patrick showed you a very nice ways of doing this using a technique that interprets the embedding space. And we propose an alternative way that is pretty general for a large class of these types of models. So all those models we're talking about, they are doing this conditional regression. So you have a ways to predict the probability of a nucleotide given a sequence context. So what we say is let's use this and now go take pieces of DNA with a fitted model and consider always pairs of nucleotides, call one a query, and perform in silico mutations, and then ask the model, how do you change, whether you change and how much, for the probability at the target positions. I'll show you how this works. Just a little warning in the following. You will see results for many more large language models than the one I showed you before, so not everything is going to be from this species LM. The first result is with our species LM in fungi. And we spotted that actually the model was pretty good at tRNAs. They ended up in our regions. And so here is an example for the tRNA arginine, where you have a very good reconstruction of the sequence. So the model has recognized this region. We know the secondary structures of tRNAs. We know that this position here, this very first base, is binding the second to last base in the structure using a Watson-Crick binding. So now we can ask the model, if this were not to be a T, but an A, what would you predict at that position? And beautifully, it predicts then a T. So it has learned, indeed, the base pairing, and not simply by heart that sequence. And you can see it for all positions. You have this Watson-Crick pattern showing up. And you can, or what we did then is we summarized this 3 times 4 matrix by this maximum number. This is the maximum absolute value of log odds. We tried other alternative metrics, but this worked pretty well. And then what we do is when we enter a region like this, we take every position as the query, every position as the target, and we record in the matrix this score, right? And so you get, for the case of the tRNA, this beautiful anti-diagonal here, which is exactly this acceptor arm, but also the other arms. You even get something more. As you know, the tRNA is not just a secondary, it's a tertiary structure, and this is achieved thanks to further bases that get the 3D. And one of these bases, there are 3 of those, but one of those contacts is also captured by this simple species LM. And just to really nail that, of course, these types of analysis were done before using a technique called covariation analysis, but it was always done in a way that you required alignment, you required to know where the tRNAs were. So everything I showed you here was done without focusing on tRNA loci. We only took these flanking regions, these regions flanking coding sequences. We didn't perform any sequence alignment. We didn't enter the Watson-Crick rules. So everything comes directly from just having seen many instances. So that's very exciting. And then we decided to study this more systematically and to generate such interaction maps across genomes. And so when you do that and you fly over genomes, you see patterns that are a bit recurrent, and you find a lot of things along the diagonal, which maybe are the reflections of mutagenesis biases or so. But you also find blocks on diagonal. You find off-diagonal blocks, I'll show you this later, and you find parallel and anti-parallel patterns. And then we looked for those a bit more systematically, and I will first show you what happens with the on-diagonal block. So for that, this still in the species LM fungi, we can look at the promoter of the gene SMT3 in yeast. And what you see if you just look at reconstruction, you see this polyase stretch here, nicely recognized, as well as those motifs which we can look up and correspond to transcription factor motifs. Now you can do the dependency maps. And these things that are a bit the same, or indistinguishable at that level, start to distinguish now if you look in the dependencies. So the base is the A's. It is very clear for such a model if you mask one A out of 20 to fill an A in the middle. But if you mutate an A, it will not much change the probabilities of A's otherwise on the other side. So that means there is dependencies, but it's very diffused. Whereas when you hit a transcription factor binding site like CBF1 here, well, changing one out of these 6 nucleotides has a huge effect on the probability of the others. So you get these block patterns here. And then what we've done is we said, okay, let's just call those blocks with a very simple convolution here. We're just calling regions that look like this. And benchmark that against experimental assays where we know where transcription factors are binding in yeast. And beautifully, you reach the same Brock curves, the same performance, than if you take position weight matrices that have been trained using ChIP-seq data. So here, this is the databases of position rate matrices for yeast, where we know the motif thanks to experimental data, and you get the same ROC curve than if you just call blocks. So that shows that, or that suggests we could use this to go for organisms or regulatory layers that are less studied. And this is much better than if you do the reconstruction, which will give you a lot of artifacts like this polyase. Whoop, this was a lot of results. Okay, where am I? Here. Okay, there we also find blocks that are interactions between blocks, and the splicing gives us a very nice examples of that. Here you see a donor site, a branch point, and an acceptor site, and there are interactions between those. Those three elements are defining together how splicing occurs, and one can use then this, we can look systematically at dependencies between a donor site and acceptors and have a way to find which are the true donor this way. Okay, so these off-diagonal dependencies are indicating which elements are functionally working together. I mentioned to you we also have anti-parallels, and this is can be also very easily called on those maps. And when we find them, we typically hit RNAs that have secondary structure. Here I'm using another model which has been trained on RNAs. So it's an RNA language model developed by another group. We only take the large language model and perform those analyses. And you see in this very beautiful E. coli RNA structure that the elements are pretty nicely found, including also pseudoknot interactions. Okay, we can do then this systematically, call all those elements here, then benchmark them against known secondary structures, and get for a large class of structured families, good layer rocks. So that shows that even without fine-tuning on secondary structure prediction, there is already, just in the statistical dependencies, evidences for those functional interactions. We contacted a group who works on secondary structure experimentally, Danny Incarnato, and he performed experiments and validated four of the new structures we found for E. coli. This is an assay called DMS, where bases that are in Watson-Crick pairing are protected from the agent, and others are reacting to it. And what we could see here is that those arms here indeed correspond to stretches that are protected from the DMS, and the pseudoknot here correspond to these bases over there that are also protected from the DMS. So that's supported by this data. We're collaborating now further with Danny Incarnato to integrate these together with algorithms that analyze DMS data to go into calling secondary structures and ensembles of structures. Okay, the last element that we can find are these parallel dependencies. So the anti-parallel were typically for reverse complementary regions as we find them in tRNA structures. We also find parallel, and there what you see is this typically relates to elements that are replicated. For example, this tandem replicate. And what is interesting from a machine learning point of view means that the algorithm, when it has to predict a base here, what it does is simply look up the identity of the base in the other one. It has not learned by heart the block. It just looks up. So it understood somehow. It has a model of replication, right? And we can show this because we can artificially put in the DNA, in silico, artificial sequences at different spacing, and then we see that as long as the sequence is long enough, like 14 or 16, the model will indeed predict properly the identity of the other bases. They were not in the training data sets. This is a phenomenon that is also known in natural language processing, but it turns out that on genome, those models are able to find that. We can also use this to benchmark without fine-tuning large language models. So in this case, for example, if you're interested in tRNA secondary structure, you can ask for different models or the dependencies recognized. So this is the RNA language model Rinalmo. This is the one, the species LM fungus. This is actually a human tRNA, but it turns out tRNA are so conserved that you can use a model trained on yeast, on fungi, and get a good structures for human tRNAs. And you see that we are performing pretty well. EVO, which is the first version of what Patrick showed you today, is also doing not too bad. There's a few artifacts due that it's a next token prediction, but we can average over the two directions. And also, it is trained on prokaryotes, and so here we're looking at a human tRNA. So it's not so bad, but and then you have other models like NTV2. This one is tokenizing the genome into pieces of six bases separated, so 6 mirrors that are non-overlapping, and that seems to introduce a lot of artifacts with dependencies that are not natural there. And then Hyena is a model that was trained in this case only in the human genome. And that's a very clear lesson from what this analysis is, that the models that have been trained only within the variations within the human genome were not able to capture those things. So going multi-species is key. Okay. So we then thought, well, if we have this, which tells us about dependencies between pairs, maybe this is also helpful to come back to this very general concept of variant prioritization, variant influence scores. And so what we propose is we take every position and we look simply at the average effect across all the other bases as a score. Okay, sorry. All right, so if you do this and you take a clean var and you define, try to distinguish benign from pathogenic, you have a pretty nice distinction. This is the variant influence score here. And it performs better than if we do using this reconstruction, the classical log ratio that is used in many other studies. This is done using a model of VAS, which is the same logic than the species LM fungi, but we applied it to nearly 500 metazoan. However, this is not beating the classical conservation scores, like phylopy, alignment-based conservation. Now, as you know, there's a lot of circularity here because variants that enter those databases have been typically found by classical tools, so perhaps it's easier for conservations. So we turn into a mutagenesis assay. This is a screen from Martin Kircher and Jay Schunder on enhancers and promoters, and I will go show you the results on the promoters that correspond to also the regions where we train our large language model. Just qualitatively, there's some kind of agreement between reconstruction probabilities and the effect on these reporter assays. However, this log ratio by itself is not beating conservation. But what we can do is we can do on the same large language model, do this variant influence score. And now we are above what conservation gives you on those 9 promoters. So this is nice. It shows that this effect indeed seems to be a good idea. And we managed to run EVO2. It's a pretty big model. I don't know if I have to send you the bill, Patrick, but okay, you can try. And it also for EVO2, going into this influence score is gives you a better correlation with the measurements than if you use the log ratio. So these things perhaps generalize to other large language models. I don't want you to understand from just 9 promoters that we are doing better than EVO2. We need more benchmarks for that. But these differences here is something we seem to see in many situations. Okay, then, this is what the self-supervised learning model field does. But if you're working into gene expression, well, we're actually against people that have done very beautiful supervised models using things like ENCODE. So how do we do against those models? Borzoi is perhaps the state-of-the-art now model, the successor of Informer in some sense. You see that it's on this promoter assay, it's not doing significantly better than this simple variant effect prediction. And it seems when doing a simple linear model trained on those 2 scores that we improve a bit. So perhaps we are bringing here some aspects that are missed by Borzoi. Again, more experiments must be done to really nail that. So to conclude these results, I think these nucleotide dependencies can be helpful for regulatory genomics, especially going into species we have not studied so well, or post-transcriptional regulation. It has a lot of potential for RNA structural biology. We also have results on tertiary contacts, etc., a bit systematic. So revisiting this field with RNA foundation model is good, and this tool can help you telling which one seems to have the right types of biology that maybe the current models are missing. It's a good tool for evaluating large language models without fine-tuning. It may give us a new way of doing variant interpretations. And then also it's a way to see beautiful pictures across the genome. I still have some time, or what is it? Yeah, okay. So in the next slides, I just want to give some reflections about maybe the future directions in these fields of DNA language model. And it's a bit this differences between variant prioritization and representation learning. And so the first thing I want to highlight, and it seems to resonate a lot also with what you guys know from protein language models, is that these large language models are learning from two sorts of evidences. On the one hand, we have the classical orthology, right? So every piece of DNA we have is inherited from the parents, and so eventually you have orthologous regions across species that descend from a common mass on that. And so the footprint that we see here is the capacity to be propagated to the next generation. So that's fitness. So alignment gives you, which is the old tool to find out which are orthologous genes, is very helpful to find fitness. But we also can learn across the genome, right? The tata motif, the path3 motifs is found in many UTRs. And so these genomes, these large language models also learn from things that are across genes. And traditionally, we had already software for this. For example, the MEME software is a software that finds overrepresented motifs within predefined regions. And we have also alignment-based conservation, which gives us scores for fitness. So that's on this axis. And what we see now is that we have large language models that have been trained on single species. They perhaps only trained that. Things like what we have been doing, or Patrick has showed you, are going more into this, like learning within and across. And then there are models that are proposed now. You would maybe heard about GPN-MSA, which is a large language model, but that takes a multiple sequence alignment as input. And so this learns different things, because here when we start from an alignment and want to predict what is the value of that base here, given everything I have that, I'm very much pulling into the fitness and the selection in this locus. I'm not necessarily needing to learn a representation of all the motifs out there in the genome. We'll have to do more investigations on that, but we've done a first analysis, again, on our tRNA of this model, GPN-MSA. You see a beautiful reconstruction. So the alignment probably tells you these bases are very much under constraint. And so if you mask one in human, just you can look it up from the other genomes. But if you do dependencies, you say, what if I mutate this? Does that change your predictions elsewhere? Not much. The position elsewhere is based on the column. So you don't really learn about the dependencies between those bases. So is GPN-MSA learning something that is helpful for representation learning, for performing tasks that are more specific? That's not clear. But I think that's something we should study more in the future. And perhaps also, depending on what is your goal, if you're doing general variant prioritization, for example, in human genetics, these alignment-based models may be good for a while, whereas if you are more interested in performing very specific downstream tasks, such as binding site predictions, you may want to take from models that are a bit more general, like what we showed or Patrick showed before. Okay, and the last point I want to highlight, we need more mutagenesis scans for non-coding and regulatory parts, and I was very happy to see some of those in the talks and in the posters, and happy to collaborate on that if you like. And to finish, I'd like to thank this team. I'm very happy, very glad to have motivated and smart people in the lab. It's been really a pleasure. And then the authors, Alexander Carolus, Johanna Singerl, and Dennis Gankin for the species LM, and then Pedro Tomás del Silva and Alexander Carolus for the dependencies. Thank you for your attention. I'm ready to take questions.